Hey guys, welcome to lesson 10 for our disease unit. This is our last lesson before your review and your test. So hopefully things are kind of coming together and you're getting kind of used to this whole e-learning situation. Today, we're going to talk a lot about Ebola, and this is another virus that caused a lot of problems, especially in African countries back in like 2016. You probably remember hearing about it on the news. And so just like HPV with Henrietta Lacks and just like COVID-19 right now, these are all kind of connected to one another. So a little bit of background information about it you can read here about where it's happening, when it was happening, tracking how the virus spreads from person to person, and how we can figure out how mutations, if we track mutations, we can track how the virus is spreading. These are some examples of that and see what's going on. So first we want to define the term mutation. Hopefully you remember this, that it's a change in a DNA molecule. So one letter or one nucleotide is wrong. And then in our own words, why is it important to examine the sequence of the Ebola virus genome? Genome is another word for DNA. During an outbreak. So I said knowing how the virus is mutating over time is important for understanding how it is spreading. It can diagnose individuals who are infected, and it can help determine whether the virus is becoming more infectious. So people who are more likely to die or pass away from infections, they might have a different mutation. So we can track how deadly it is. Okay. All right. So then I know that this link didn't work, and I'm really sorry about that. I thought it would, but then it didn't, so I put the links that you need for this lesson in the attendance page. So go back and look at the attendance sheet for like the last day I took attendance, and you can find those links there, okay? So what you wanted to do is essentially you just wanted to group them. However you wanted, you can move the sequences around the slide and put them into groups however you wanted. So here's what I did. You could do whatever you wanted, but here's how I did it. As long as you can justify why you put things in different groups, I don't care how you did it. Okay, so here I had this group which had only these two mutations in it. And then in this group, we had these two mutations and then random other ones. And then in this group, all of them had the TC, and the AC, then had a couple of randos too. Okay, so that's just how my brain worked. Whatever your brain did is fine too. So describe the criteria or the method you use to put your sequences in different groups. A couple things you could say. You could say identical sequences, sequence that only differ by one mutation, the mutation location, so which number, the number of mutations total, or sets of mutations. Okay, so however you decided to group yours is fine. And then if you could think of another way to group them, you can put that here too. Okay, so it says if a sequence has a larger number of mutations, does that mean it is from earlier or later in the outbreak? And so it's definitely from later in the outbreaks because mutations accumulate over time. So the more mutations, the later in the outbreak it was collected. So more mutations happen after a longer period of time. All right, then it's a creative visual that shows the relationships between the groups you made. So a lot of people had some issues with this one. I said you could do it on a regular piece of paper if you wanted. You could draw it here. Here's a good example for it. So for the part 2 procedure, it shows you, so here we have time, here we have our reference sample, and then kind of the changes that happen between each one. So if I was to go over here, sorry, that was a leftover. If I was to go over here and look at mine, okay, so I have the reference sample first, just like they did before. And then I know since we have more mutations over time, I would do this one first because it only has 2 mutations. So I'd have I'd have sequences 4, 6, 11, 13, and in between we just had 2 mutations at spot number 2 and spot number 6. So here's 2 and here's 6. Then I would do this group. So then I would have the next group is going to be 3, 1, 9, 14. And then they have other random mutations, but they still have these ones too. Okay. And then the last group, we've got 2, 10, 15, 5, 7, 12, 8, and they have their additional mutations are at 1. So they have 1, 2, 3, 4. 4 new mutations at 1, 7, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. Okay, and so then if I go over time, I'm going to know that time goes this way. So these ones here are the youngest viruses, and these ones here are the old, or no, sorry, that's backwards. These are the oldest mutations, and these are the youngest, or the more recently mutated. Okay. All right. Then in the next one, I put them into groups for you already. And so this is how the scientists at the hospital put them into groups. All right. So you can see that we've got the same base core mutations, and then we have a few randoms in between. Okay. So basically for number 7, you're just going to compare and contrast. So it just depends upon what you're doing. And then you got to look for the differences. So the difference between the reference sample and the group 1, we have C in position 2 and T in position 6. Between 1 and 2, we have T in position 1, C in position 7, A in position 12, and C in position 13. And then the last one is C in position 14. All right, so if we had to put them in order for who's the oldest or who's the youngest, we're going to have the reference is the oldest, then this one, then this group, then this group. Okay, so these are the newest mutants right here. Okay, explain how the sequences support the hypothesis that mutations accumulate over time. You're gonna be able, it definitely shows that, it's definitely true. We know that mutations in one virus will be passed on as it replicates and infects other people. So the additional mutations as they replicate are passed on and so on. And then, but what happened too is like, how can we explain that these randos did not get passed on to the new groups, that these ones were only present in 1 or 2 sequences? How do we explain that? And if you think back to the mice, when we were talking about how mutations are super random and some of them are beneficial to survival and some of them are not beneficial to survival. So any mutations that maybe stopped survival of the virus, maybe it was a quote unquote bad mutation or harmful mutation that led the virus to quickly die, they couldn't pass on those genes, so the mutations wouldn't move on to another groups. However, if it's a beneficial mutation, then everybody after that mutated one, if it's a beneficial virus, is going to have it. Okay, then there's the extension activity. Again, this is in the attendance section for the link. Sorry about that. And you just got to go through and look at your Ebola virus. I also found this cool picture of a helical virus shape, because a lot of people think that all viruses look like weird spiders, and they don't. This one is helical, and I thought that was cool because DNA is a double helix. So how cool is that? All right, for our vocabulary, we've got host, transmission, vaccine, and zoonotic. And then for our wrap-up and review question, what's Ebola? Ebola is a disease caused by the virus that causes fever and severe internal bleeding, usually leading to death. How can tracking mutations help us understand the evolution of a virus? When we look at the genetics, we can see how it changes over time and track who got the virus from who. Couple of questions. How does this relate to the Ebola virus that we read about a few years ago? Definitely. How does this relate to the COVID-19 viruses that we are experiencing now? And then you want to write down a new question. Okay, our next thing that we're going to do, we're not really going to do a new unit, but we are going to do something called biobattles, where we're going to look at your immune response to viruses. So here's a little background about that and what's going on in your immune system. And so here's how a virus gets into the cell. So it attaches to the membrane, it kind of opens up and then kind of like dumps out its DNA and all of its virus parts into the host, and then your cell is officially infected. Then it says, why is the virus that infects humans not as likely to infect a dog or a cat? Well, dogs have different types of cells, and maybe the virus doesn't recognize it, so the puzzle piece doesn't fit. Eventually, the host's immune system catches up and helps with the spread of the virus, which is why vaccines are so important. Okay. After the DNA is released into the host cell, the virus DNA integrates or goes into the host DNA so that the host DNA can help make copies of the virus's DNA. So remember, viruses can't make copies of their own DNA, so they need the host DNA to kind of help them make copies of themselves. Okay, and so once it makes copies of itself, it can spread to more cells. So super creepy, but also awesome. That's all I've got for this lesson. See you next time.